So every one of your cells has about 6 billion letters of DNA, A, C, G, and T. Your genome, which is the sum of these 6 billion letters, is not perfectly stable. Mistakes can be made while in development as an embryo. Mistakes can be made when your cells replicate as you grow or as you heal injury. If you smoke, get exposed to sunlight, eat certain kinds of foods, some of those mistakes can lead to diseases if they're not corrected. My name is David Liu. I'm a professor at Broad Institute, Harvard University, and HHMI. There are 60 million children that will die before they reach their 5th birthday because of their genetic disease. These genetic diseases include thousands of different diseases. You've heard of them: cystic fibrosis, Tay-Sachs disease, sickle cell disease, Rett syndrome, progeria, spinal muscular atrophy, and on and on. There are no treatments that actually fix the root cause of the disease, the misspelling in the patient's DNA. A hallmark of chemistry is using our ability to change the structure of a molecule in order to impart new functions on that molecule. The molecule that our lab was most interested in treating, like the subject of a chemistry problem, was DNA. That eventually led to the creation of some of the gene editing tools like base editors and prime editors. Base editing and prime editing allow us to change DNA sequences of our choosing into sequences that we believe will offer some therapeutic benefit to patients. Base editors are sort of like DNA pencils. They rewrite an individual DNA letter, inverting it by actually rearranging the atoms on the DNA base. Base editing builds on CRISPR-Cas9 to cut DNA sequences. But unfortunately, for most genetic diseases, the problem that causes the disease is that the gene is already messed up somehow, and we need to fix it rather than further disable it. The idea behind base editing began in late 2013 with Alexis Comore. We first had to determine if we could recruit programmable DNA binding proteins like CRISPR-Cas9 that perform chemistry on DNA and not unwanted changes elsewhere. And once we did that, we ended up with sort of a mess. We got a machine that could change one base to another base, but it did so not cleanly at all. It was more like a base randomizer, not a base editor. It also worked very inefficiently. We thought about the problem and realized that our own cell's DNA repair machinery was making a mess of the target site that we were trying to edit. So we added this extra protein that protected the base edit from being tinkered with by our cell's DNA repair protein. The base edits in that second generation became much cleaner and much more efficient. And then finally, in sort of the third stage, increasing the efficiency of the editing. DNA have 2 strands. When we made the first base editor, we disabled its ability to cut both DNA strands. What we should do is restore the ability to cut just one DNA strand. If you nick the unedited strand, that tricks the cell into remaking that part of the unedited strand, using the edited strand as a template, which then makes the edit permanent because now both DNA strands have the edited information in it. And now pretty much all base editors use this approach. Prime editors are another kind of engineered molecular machine, but instead of performing chemistry on an individual DNA base, a prime editor acts as a sort of DNA word processor that can perform true search and replace genome editing. Prime editors are even more complicated molecular machines than base editors. Andrew Anzalone, who led the development of prime editing, broke down the problem into a series of 4 challenges that we glibly nicknamed the 4 small miracles. The first of those challenges was showing that a sequence in the guide RNA could actually be copied into a target DNA sequence. So that was sort of challenge number 1. But the cell is a very different place compared to a test tube. To address the second challenge, Andrew used yeast cells. But that was really still the early stages of prime editing. And so the third challenge was figure out how to get prime editing to work in a human cell. And that's where we encountered our first incredibly discouraging result, because when Andrew took the same system that worked so beautifully in yeast cells and tried to perform the first prime edit in human cells, he got 0 editing. When you get 0% of the result that you want, it's very hard to know if your assumptions are flawed. We made a big list of all of the possible reasons why prime editing might have failed in human cells. And it was a sobering list because some of those explanations were not fixable by us. But fortunately, the problem turned out to be an aspect that we could fix. The length of the runway of DNA that you need in order to prime the DNA copying event. This is actually why prime editing is called prime editing. That requires a short DNA runway to serve as sort of a landing strip for a new piece of DNA to begin copying the new edited DNA sequence. And that priming process is different in human cells compared to yeast cells. The runway needed to be a little bit longer. Not too long, not too short. It had to be just the right Goldilocks length. When he reached that Goldilocks length, we observed the very first prime editing in human cells. But it was not efficient. And so in the fourth challenge, we introduced specific changes to the prime editor protein that increased its efficiency enormously. And we also used the same trick that Alexis developed of nicking the non-edited DNA strand. And the combination of those improvements resulted in the first efficient prime editing in living human cells. And since then, prime editors have been used by literally thousands of laboratories around the world. And recently, the very first prime editing clinical trial has begun. One of the really exciting implications of the versatility of prime editing, we can replace just about any DNA sequence in a human cell and perhaps in a patient with just about any DNA sequence of our choosing. That flexibility opens up an enormous number of possibilities beyond simply fixing a mutation that causes a specific disease. One direction that our lab is vigorously pursuing now that takes more creative advantage of this flexibility is to perform prime editing in ways that allow a single prime editor to treat not just one mutation that causes one disease or even 10 mutations that cause one disease, but perhaps hundreds of mutations or thousands of mutations that cause many different diseases with one drug, one composition of matter, one prime editor, one guide RNA. What we call disease-agnostic therapeutic genome editing. Hopefully we'll report our first example of doing so later this year.